My dear student friends, in today's lecture, let's study the diagnosis of silkworm diseases. Sericulture is a life-sustaining occupation for a large number of people in various parts of India. Indian sericulture comprises of mulberry, tasar, muga and eri culture being practiced at different agroclimatic zones. The crops are prone to various stresses which brings down the productivity and profitability of the occupation. One of the major stresses which deter the prospect of sericulture activity is the incidence of silkworm diseases caused by various pathogens, namely bacteria, fungus, virus and microsporidia. These pathogens individually or in combination can damage a crop. The early infection during first to third instars lead to disease episodic in the later instars, inflicting heavy loss to the rearers. There are several reports of loss due to diseases in silkworms in India. The damage due to diseases varies from a range of 15 to 40%. A total loss of 10% was reported in Japan during the 60s. During 80s, the loss due to diseases has been significantly reduced to 15 to 20% in India. This is attributed to the improved rearing and disease diagnostic and management technologies. Despite after adopting personal and rearing hygiene, disease occurrence during silkworm rearing is common. The diseases found in silkworm crops result in the economic loss to the farmers, affecting quality cocoon production. The silkworm is very fragile and has lost many of its natural instincts to combat adverse conditions due to the domestication over centuries. They are highly susceptible to many viruses, bacteria, fungus and microsporidian spores. Conventional microscopy is widely used for the detection of polyhedra, bacteria, hyphal bodies, fungal spores and microsporidian spores. By the time the microscopic or visual observation is made, the disease must be at an advanced stage of infection. This would lead to the spread of diseases to episodic condition. For the detection of BM-IFE or BM-DNV, electron microscopy or immunological techniques are employed, which in normal course is impractical and non-viable. Further, the commonly employed serologic tests to identify microorganisms are not very specific to distinguish closely related serotypes. Since the control of silkworm diseases are met with limited success, early detection and identification of pathogens are important in preventing the spread of diseases to ensure safe and sustainable sericulture. Sensitive detection of pathogens prior to damage helps in effective disease management. In apiculture, poultry, animal husbandry, pisciculture, etc., the disease diagnosis and remedial measures are more effective because of their longer lifespan compared to that of silkworm larvae. At present, the major disease management strategy is the exclusion method, wherein a germ-free environment is provided by disinfecting the rearing rooms. Timely diagnosis of disease and etiological agent causing the disease is of primary importance for managing the diseases. For better understanding, the topic is being explained into different subheadings, and they are: 1. Signs and symptoms, 2. Disease diagnosis, 3. Symptomatic diagnosis, 4. Laboratory diagnosis, 5. Microscopy examination, 6. Immunodiagnosis, 7. Molecular diagnosis and finally 8 on the management of silkworm diseases. Let us study these aspects one by one in detail. The first aspect is signs and symptoms. The ability of microorganisms to enter the body of susceptible host is the infectivity and its effect is infection. The infection generally results in detectable effects in the form of injuries, characteristic aberrations or dysfunctions. These are designated as signs or symptoms. If the effect is physical or structural abnormality, it is the sign. A sign may be in the form of abnormalities in the morphology or structural changes in color, malformation of appendages, indegments, etc. The effect is symptom if it is functional or behavioral disturbance. In case where the effect is in the form of both sign and symptom, the term syndrome is used. The syndrome may be very characteristic and specific for a disease caused by a pathogen. However, the syndrome is more often due to several causes. The flachere in silkworm is represented by flaccidity, and diarrhea is a syndrome. Environmental factors, bacteria and viruses may also cause the syndromes. The second aspect of our study is disease diagnosis. Diagnosis is the process by which one disease is distinguished from the other. A proper diagnosis involves the study of etiology, symptomatology, pathogenesis and epizootiology of the disease. The importance of diagnosis in insect pathology lies in the fact that it is essential to know the nature of the disease before the disease can be properly studied, controlled or suppressed. One of the most common types of diagnosis is the differential diagnosis, wherein the signs and symptoms and post-mortem changes of different diseases are put to comparison and contrast in a systematic manner to distinguish one disease from another. Sometimes preliminary diagnosis is carried out, wherein the first examination will be made. It proposes possible type and causes of the disease, which is almost a guess. Tentative diagnosis is made after microscopic and macroscopic examinations. Definitive diagnosis is the complete diagnosis, wherein a final conclusion is drawn after gathering all the pertinent data and its analysis. The conclusion is based on the information derived from field, laboratory and literature. There are various types of operations in diagnosis, such as symptomatic diagnosis, laboratory diagnosis, post-mortem diagnosis, etc. The third aspect of our study is symptomatic diagnosis. It is generally found that the infected silkworms behave differently than that of the normal uninfected ones. Therefore, observation of the silkworm colony for abnormal behavior is very vital to differentiate or diagnose disease infections. Usually, symptomatic diagnosis is resorted to the diagnosis of diseases, but many disease symptoms are atypical with similar signs and syndromes. Therefore, after preliminary behavioral observation, laboratory and post-mortem diagnosis becomes inevitable for correct diagnosis of the problem. The symptomatic diagnosis primarily involves general inspection for typical symptoms. Visually different silkworms such as underdeveloped larvae, delayed or failed to molt larvae, worms with lethargic movements, worms with poor appetite, etc. could be signs of pathogen infection. Such larvae are definite candidates for laboratory diagnosis too. For the diagnosis of different silkworm diseases, the key diagnostic feature of each disease will help, and let us see them one by one. Bacteremia. The silkworms with the bacteremia or the bacterial disease of digestive organ, the cephalothoracic region becomes translucent and develops diarrhea. The larvae lose appetite, become sluggish and show retarded growth. Large numbers of cocci bacteria are observed in the gut while conducting microscopic examination. Bacterial septicemia. In the case of septicemia, the larval body straightens, becomes shrunken and the thoracic region swollen. The larvae die in short period. Several bacteria such as Serratia marcescens, Pseudomonas species, Streptococcus species, etc. are observed in the hemolymph. The body of diseased silkworms becomes soft, discolored, and the body will rupture easily, liberating foul smelling fluid containing large number of bacterial cells. In case of septicemia caused by Bacillus species, the silkworms shortly after death develop dark greenish tinge. But in case of Serratia marcescens, the larvae after death develop dark brown spots and reddish tinge. Bacterial toxicosis. The larvae lose appetite and become sluggish. The dorsal vessel pulsates at rapid rates and larvae wriggle. The larvae lift head up, develop spasm and tremors and vomit gut juice, paralyze, collapse and die within a short span of time. Nuclear polyhedrosis or grassery. The signs and symptoms during initial disease development will not be apparent. In the adult silkworms, the symptoms appear 5 to 7 days post infection. The larvae fail to molt or at times imperfect molting is also observed. The intersegmental membrane are swollen and the worms appear restless. The hemolymph becomes turbid and milky due to the multiplication of polyhedral bodies. The skin loses its elasticity, becomes fragile, gets ruptured easily and milky white fluid oozes out. Cytoplasmic polyhedrosis. Affected larvae exhibit symptoms of slow growth, stunted and flaccid body, irregular molting, reduced mulberry consumption, vomiting and dull white body color. The fully diseased midgut appears opaque and milky white. Densonucleosis. The infected larvae may look pale and exhibit stunted growth and empty headed. Head and thorax are upheld motionless, feces irregular shaped or diarrhea may exist. The gut is filled with brownish fluid. The body flaccidity is a major sign. The alimentary canal appears pale yellow with little internal contents. Infectious flatury. The worms feed sparsely due to loss of appetite. Infected worms look sluggish and sicky with the head and thorax upheld motionless. They are empty headed and exhibit flaccid conditions due to loss of elasticity. Since thoracic and abdominal portions are stunted in growth, the heads of the infected worms appear to be disproportionately enlarged. Chain or string form of excreta is observed. Shrinkage of body and rectal protrusions are common. White muscardine. Sluggishness of the worms and increased rate of dorsal heartbeat are observed. The larvae become inactive and pale in color. The area surrounding spiracles and intersegmental parts turn brown and later become black spots. Vomiting and diarrhea are also observed. The larvae stretch the bodies upon litter and become soft to touch. White efflorescence covers the whole larvae and become mummified with the fungal conidia. Green muscardine. Except the green color of the mummified larvae, all other symptoms are similar to that of white muscardine. Aspergillosis. Young silkworms are susceptible to this disease. The body of invaded worms become compact and lustrous and the larvae die soon. The body becomes hard around the site of infection. Mycelial cluster develops quickly on dead worms. The color of the conidia varies with the species or fungi involved. K. Microsporidiosis or pebrin. First instar larvae infected embryonically show no spacing of body hairs. 2 days after hatching, the worms become darker, thin and exhibit slow growth. Such larvae will not survive beyond third instar. The larvae infected at later stages show incomplete molting, difficulty in molting, delayed molting, etc. Unequal size of worms in a colony is the striking symptom. The homogenate of infected tissues may reveal microsporidin spores under microscopic observation. The fourth aspect of our study is laboratory diagnosis. The laboratory diagnosis covers the microscopic and macroscopic examinations for pathogens, its study, assay of non-microbial factors, biochemical and biophysical tests of tissues and fluids, and the observation on the course of disease. Pebrin, nuclear polyhedrosis, cytoplasmic polyhedrosis, bacterial flatury and muscardine could be monitored and diagnosed following microscopic examination at the early stage of infection. However, certain pathogens such as BM-IFE and BM-DNV require specialized diagnostic approaches, as the symptoms of the disease caused by these are atypical at early infection stage and the pathogens are submicroscopic. The immunodiagnostic and molecular techniques are followed for diagnosis. These tests have also been employed for the detection and discrimination of different strains of pathogens. Then post-mortem diagnosis. Post-mortem diagnosis is the test conducted after death due to disease, similar to laboratory diagnosis to ascertain the cause of death. The fifth aspect is microscopic examination. Microscopic examination of homogenate of eggs, moth, pupae and larvae for pathogen or its developmental stages is an appropriate approach to monitor diseases. It is Louis Pasteur who proposed the microscopic examination for detection and elimination of microsporiosis. He suggested the microscopic examination of egg laid by moths for microsporidian spores and advocated the rejection of eggs laid by such infected moths to purge the silkworm generations from infection. This was a turning point in the history of sericulture development and saved the industry from disaster. The bacterial, fungal and polyhedral infections could be diagnosed by microscopic examination. Sample smears of various stages of silkworms are tested for possible disease infection. This helps in the management of silkworm diseases. The sixth aspect of our study is immunodiagnosis, which is very significant. The symptoms of many silkworm diseases are atypical and microscopic observation of some pathogens are not possible as they are submicroscopic. Immunodiagnosis becomes a handy tool in identifying the disease. The detection of antigen-antibody reaction complex forms the basis of immunodiagnosis. The antibodies react with antigen to form insoluble complexes such as lattices that visibly precipitate from solution. This phenomenon is the basis for many classic serologic and diagnostic assays such as precipitin test and co-agglutination test. Let us study various immunodiagnostic tools used for disease diagnosis in silkworms. Precipitin test. The antigen antibody reaction, if it occurs in liquid media in test tubes of smaller diameter, a precipitin ring is formed. This test in a tube is ring precipitation test. Immunodiffusion. Antigen antibody reaction is done by introducing antigen and antibody into different regions on an agar gel and allows them to diffuse towards each other. They form a band of precipitate at the junction of their diffusion fonts. Immunoelectrophoresis. In case of complex mixture of antigens, the antigens are subjected to electrophoretic diffusion. The resolution of the reaction band is improved by adopting this method. Even though the precipitin and gel diffusion tests provide unequivocal evidence of specific antigen antibody reaction through the formation of precipitin ring or line, they are least sensitive, requiring comparatively larger quantity of antibody. In addition, some antigens are too large to diffuse through agar gels. Co-agglutination test. Generally, the macromolecular antigen and antibody are mixed in liquid medium. The macromolecular antigen specifically clumps or agglutinate, resulting in co-agglutination. It becomes visible to naked eye or under low magnification. Agglutination test. In case of submicroscopic antigen, the co-agglutination takes place but remain invisible to naked eye or at low magnification. However, it is possible to absorb the antibodies onto particulate labels such as latex polystyrene beads, bacterial cells, red blood cells, colloidal gold, ferritin, etc., which gives visibility to antigen antibody reaction. antibody adsorbed onto latex polystyrene beads, that is 0.65 to 0.85 micrometers, are used as test reagent in diagnosis. Pallas test. The above said adsorption of antibody to the beads could be improved by pre-coating the beads with protein A. The test is performed following the latex agglutination test procedure and referred as protein A linked latex antisera, that is Pallas test. The method economizes the use of antibody and improves the sensitivity. Radioisotope labeled immunodiagnostic assay. This technique makes use of a radioactive isotope which are conjugated onto antibodies without deleterious effect on the immunological activity. The bound label can be detected by a counter appropriate to the isotope or a photographic film or emulsion. Enzyme labeled immunodiagnostic assay or ELISA. The assay uses enzyme as label to signal the occurrence of antigen antibody reaction. It is a heterogeneous immunoassay involving steps during which the reacting and non-reacting compounds are separated. The reaction takes place on a microtiter plate to which reactants are sequentially presented and successively bound. After each step, the non-reacting and inhibitory components that remain unbound are washed off. Finally, a substrate which reacts with the enzyme to develop colored substance is presented to reveal the occurrence of antigen antibody reaction. Enzyme based dipstick immunoassay. This is based on the principle of ELISA. The dipstick immunoassay uses nitrocellulose membrane strip as test matrix instead of polystyrene coated surface. The specific antibody is captured as dot on the nitrocellulose strip and the remaining surface is blocked. Colloidal dye based dipstick immunoassay. This is much simpler than enzyme based dipstick immunoassay. The assay includes the dipsticks prepared as above and only one test reagent. The test reagent contains specific antibody adsorbed onto colloidal dye particles. The homogenate of the sample is incubated in the dipstick for 30 minutes and after wash in water. Incubate in test reagents containing specific antibody dye particles by dipping for 30 minutes. In positive samples, colored spots are developed. Fluorescent antibody test or FAT. This test combines the specificity of an immunological reaction with high sensitivity obtained from fluorescence emission from fluorescence marker such that the site of interaction between antigen and labeled antibody can be observed under fluorescence microscope. The seventh and final aspect of our study is molecular diagnosis. The diseases present serious problems in terms of diagnosis and prevention as infected animals often do not demonstrate clinical signs until there is advanced disease and infected animals appear to be a constant potential source for transmission. Detection of the presence of disease causing pathogens and their early detection in a silkworm colony helps in better managing the disease in silkworms. The presence of a pathogen in test material can be identified through the detection of the nucleic acid of the pathogen using different molecular techniques. Restriction fragment length polymorphism, that is RFLP. Restriction fragment length polymorphism analysis, that nucleic acid fingerprinting, can detect differences in the genomes of closely related microbial species. To map the genome of a microorganism, the DNA is extracted and clipped into fragments of specific nucleotide sequences with a restriction in endonucleosis. The resultant DNA fragments are then separated in agarose gel by electrophoresis and visualized with ethidium bromide. The RFLP analysis may be used to determine the differences or similarities in the genomes of different virus isolates belonging to the same serotype. In this way, the involvement of a particular virus isolate in a disease outbreak can be documented. Polymerase chain reaction, that is PCR. The polymerase chain reaction procedure exploits natural DNA replication, but produced in vitro. Large quantities of a desired sequence of DNA are made from a complex mixture of heterogeneous sequences. The amplification of DNA by the PCR is accomplished via a succession of incubation steps at different temperatures. A novel PCR based assay for individual and simultaneous detection of microsporidians, nucleopolyhedrovirus and densovirus infecting the silkworms is available. The assay has high specificity and sensitivity for the pathogenic DNA. This method is a valuable supplement to the conventional microscopic diagnostic methods and can be used for the early detection of pathogens infecting silkworms. Furthermore, it can assist in the safe supply of disease-free silkworm eggs to the farmers. Thus, the topic can be summarized that the silkworm colonies under controlled conditions are susceptible to various diseases caused by bacteria, virus, fungus and microsporidians. The diseases caused by these pathogens are manifested in the form of various symptoms, but quite often the symptoms are atypical and the cause of disease may not be understood properly. But management strategies to arrest the spread of diseases varies with the pathogen involved. Therefore, it becomes essential to diagnose the disease correctly, that too at the early stage of infection. Adopting an intensive approach towards identifying the diseases and the pathogens involved is significant to achieve successful crops. The diagnosis is possible by adopting visual, microscopic, immunological and molecular techniques. Thank you.